Greetings. I would like to first of all thank the organizers for the interest in my research and the opportunity to present at this meeting. My name is Shirley A Micallef and I am an associate professor in the Department of Plant Science and Landscape Architecture at the University of Maryland, with a joint appointment with the Center for Food Safety and Security Systems. The title of my talk is Food Safety of Cucumber, Field Production, Cultivar and Fruit Waxing. Today, I will talk to you about the microbial safety of cucumbers and how production practices, post-harvest practices, and inherent traits of cucumber can affect the associated microbiota and enhance the microbial safety of this vegetable. Cucumber consumption in the United States is estimated at about 3.5 kilograms per capita annually, and this consumption has remained consistent over the past 6 years. On the other hand, cucumber production in the United States has declined, and the volume of imported cucumbers has increased, especially as more specialized varieties have come on the market. The share of imported cucumber has shot up steadily from under 10% in the early 1970s to 70% and more in the past 5 years, which comprises over 900 million kilograms of imported produce. Unfortunately, several foodborne illness outbreaks with Salmonella enterica have been linked to both domestically grown and imported cucumbers. In the period 2012 to 2018, in the United States, there were 15 outbreaks that were identified, totaling 1,444 confirmed illnesses and 310 deaths. These were caused by multiple serovars of Salmonella enterica subspecies enterica, namely Salmonella puna, Salmonella Newport, St. Paul, Joviana, Oslo, 4512I, and Salmonella infantis, and Salmonella Thompson. The serovars causing the most outbreaks were Salmonella St. Paul and Salmonella Joviana, with 4 outbreaks each. However, these were not the outbreaks with the most illnesses. The most illnesses were attributed to Salmonella puna, which was the largest cucumber outbreak reported with 907 confirmed cases, followed by Salmonella Newport with 296 cases from 2 outbreaks. The serovars that caused the highest proportion of fatalities in relation to number of cases were Salmonella infantis, where 29% of cases resulted in death, and Salmonella Joviana, where 26% of cases led to death. Over the past 7 years or so, we have been conducting research on how cucumber field cultivation practices influence the microbiota associated with cucumber fruit. We have been assessing how differences among cucumber varieties and their morphological traits may impact association with Salmonella enterica. And we have evaluated the post-harvest practice of waxing fruit as a risk factor for food safety. Very little data exists on the effect of cultivation practices on food safety risk for cucumber. In the United States, cucumbers are typically grown horizontally over a mulch cover, with fruit touching the ground as they grow. But this vining plant grows very well in vertical systems. So to assess whether a production system affects bacterial dispersal onto leaves, flowers, and fruit, we conducted field trials over 2 years. And in these trials, we grew cucumber cultivar Market Moore 76, which is a commonly grown commercial cucumber, either horizontally, as is traditional, on plastic mulch, or straw mulch, or bare ground, so just soil, or vertically on trellises installed on bare ground, as you can see in the picture. The soil that in this field had been previously amended with raw manure. So then we collected soil, leaf, flower, and fruit samples over the growing season to quantify the bacterial indicators of food safety, E. coli and Enterococcus species, as well as to assess bacterial dispersal in the system. These bacterial counts on flowers and fruit are representative data from year 2 of our trial. We found low levels of E. coli, both on flowers and on fruit, and only during the first sampling week. There were no statistically significant differences in levels of E. coli by production system, that is vertical or horizontal production, and mulch type. We did enumerate substantial levels of Enterococcus, which is expected as several species of Enterococcus are associated with plants. We found that levels tended to increase over time, especially in fruit. We also noted that in the first sampling week, levels of Enterococcus on fruit grown on straw mulch were higher than levels recovered from fruit grown on bare ground. But no other differences due to the type of mulch or production system were detected. To glean information about the dispersal patterns of E. coli in our system, 127 E. coli isolates, of which 24 were recovered from fruit, 44 from flowers, and 59 from soil, were genotyped by box PCR to assess relatedness among strains. Here you can see the hierarchical cluster analysis conducted with those data. The brown circles, yellow diamonds, and green rectangles in the dendrogram indicate soil, flower, and fruit recovered isolates respectively. The colored bar below the dendrogram shows the ground cover and vining habit, so straw, bare ground, or plastic with horizontal habit, or trellis on bare ground with vertical habit. Numerous related isolates from soil and flowers on bare ground, straw, and trellis beds were detected, suggesting that vertical production did not present a barrier for E. coli dispersal to the crop. Modes of bacterial dispersal between soil and flowers could include wind and rain splash, especially since many isolates came from bare ground. None of these were related to isolates recovered from fruit, evidence that flower colonization did not necessarily persist to the fruit stage as surface colonization on the fruit. One cluster contained isolates from flowers and cucumbers but not soil, indicating a source other than manure-amended soil, such as, for example, an insect vector. Straw may have been a source of E. coli, as a number of closely related E. coli isolates were retrieved from soil and cucumber fruit from straw mulched beds. The second part of my talk will present data on how various cucumber varieties interact with Salmonella enterica. There are several varieties with fruit that differ in size and shape, chlorophyll content in the cucumber skin, morphological features on the skin such as ridges and bumps, as well as presence, density, and type of trichomes or spines, and possibly also phytochemical composition. There are varieties that are predominantly grown for pickling, while others are grown for fresh eating. For example, Diamant is a variety that can be both pickled or eaten fresh. The fruit of these cultivars were used in an inoculation assay with 5 different Salmonella serovars that are listed on the left-hand side of the slide. Here, the data are pulled together by serovar for the 10 cultivars tested. The y-axis is showing log reduction in Salmonella cell number that is retrieved in relation to the inoculum after 24 hours of incubation at room temperature. There is a differential favorability of cucumber varieties to Salmonella. The cultivar Excelsior exhibited the least log reduction, therefore supported Salmonella the most. This was significantly different to 8 other cultivars that exhibited a higher log reduction. The least favorable cultivars for Salmonella persistence were Market Moor 97, Diamant, and Summer Dance. A differential serovar association with cucumbers was also detected. The serovar St. Paul, Salmonella St. Paul, was the most successful serovar with the lowest log reduction after 24 hours versus Salmonella Joviana and Salmonella Newport with the highest log reduction. Although these were significantly different, the discrepancies, however, were not very remarkable, which was in fact less than half a log difference. But this is pooling all of the cultivars together. Separating out the data by both cultivar and serovar revealed differences in the cultivar by serovar interaction. In general, Market Moor 97, Diamant, and Summer Dance had the highest log reduction in all cultivars, about 2 logs in all serovars tested. Excelsior had the lowest log reduction with all the serovars. Serovars Joviana, Puna, and St. Paul also exhibited less than 1 log reduction on cultivar Piccolino. To assess cucumber surface persistence over time, many cucumbers were surface inoculated with Salmonella Newport and Salmonella Puna, and recovery was done at 24-hour intervals over 4 days. After 2 hours of inoculation, a difference in retrieved cells was detected between Salmonella Newport and Puna, with more cells recovered from Salmonella Puna. Although both serovars appeared to exhibit some growth over 24 hours, a decline was seen for the following 2 days. However, both serovars displayed a rebound on day 4 at 96 hours, and both persisted for the duration of the experiment at equivalent levels, despite the initial difference at 2 hours. We also assessed the possibility that Salmonella cells internalized into the fruit skin. This could happen through stomata that are present on the fruit exocarp. Once again, we detected less log reduction with Salmonella Puna relative to the inoculum and compared to Salmonella Newport. In this case, the cell counts, though, for both serovars, however, maintained fairly consistent numbers for the duration of the experiment, and both serovars were not only able to internalize into cucumber exocarp, but also persist for at least 4 days. No differences in cell counts were detected between the serovars or over time from 24 hours onwards. So we definitely saw internalization and persistence at equivalent levels between the two serotypes. Finally, I want to share data on the effect of fruit waxing on Salmonella persistence on cucumber fruit surfaces. Fruit waxing is a common practice. It is basically coating produce in an edible matrix of lipids and polysaccharides, and this post-harvest practice is employed to extend shelf life of fruit. Many fruit are subjected to this post-harvest practice. The main function of commercial waxes is to prevent water loss from fruit and slow respiration rates in order to slow the decay process and maintain fruit quality. Before waxing, fruit generally undergoes a sanitizing treatment, but this step may not inactivate pathogens that are strongly adhered to the surface of fruit, such as enteric pathogens, or they may be introduced during the sorting and waxing process itself, as the cucumber are transported over rollers and brushes. So this study, in our case, was supported by the Maryland Department of Agriculture following a salmonellosis outbreak that was tracked back to a packing house that was sorting and waxing cucumbers. And you can see pictures of the roller brushes that were used to wax the fruit. We conducted an assay of surface inoculation of fruit with Salmonella Newport, followed by waxing, and found that waxing favored Salmonella Newport persistence, with higher populations retrieved from waxed fruit. We did this on 6 different cucumber cultivars and saw the waxing effect on all of them. And in fact, data from 5 out of the 6 cultivars were statistically supported. We then evaluated the data in relation to the presence or absence of trichomes. So glabrous fruit would be smooth-skinned cucumbers with no spines, and on the right-hand side you can see a picture of a cucumber with spines on the fruit. Spines are also known as trichomes. And we found that higher Salmonella populations were retrieved from smooth cucumbers than cucumbers with trichomes, regardless of waxing. But in both glabrous and trichome varieties, waxing favored Salmonella on the fruit, Salmonella persistence on the fruit. This brings me to the end of my talk. In summary, field production practices such as different mulches, ground cultivation, or vertical vining did not have much impact on cucumber fruit microbiota. Cucumber varieties had a differential ability to support Salmonella association. Different Salmonella serovars exhibited varying ability to colonize cucumber fruit in a cultivar-dependent manner. Salmonella can persist on cucumber fruit surfaces and inside fruit skin for at least 4 days, with populations increasing on the surface and persisting without decline inside the cucumber exocarp, the cucumber skin. And the practice of fruit waxing was found to favor Salmonella persistence if contamination of fruit was already present. I would like to thank our funders, USDA, the Maryland Department of Agriculture, and the Joint Institute for Food Safety and Applied Nutrition. and our collaborators, Dr. Jay Zhang at the FDA and the late Diana Baldwin at MDA. And finally, the researchers who conducted this work under my supervision, Dr. Shivaranjani Pagadala, Dr. Brenda Croft, and Ms. Mary Teresa Callahan. Thank you for your attention. If you have any questions, please do not hesitate to contact me at the email address given on the bottom right. Thank you.